Annihilation.
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 Annihilation!
1 minute at a time.
I choose to love you in silence, for in silence I find no rejection. I choose to love you in loneliness, for in loneliness no one owns you but me. I choose to adore you from a distance, for distance will shield me from pain. I choose to kiss you in the wind, for the wind is gentler than my lips. I choose to hold you in my dreams, for in my dreams you have no end. Often credited to Rumi, but probably not. In the script, this scene with Lena and Kane comes later, as the transition into the shimmer, and it takes place outside. Exterior, the sky, day, the full moon hanging in a clear blue daylight sky. That same image that distracted Lena as she taught her medical students. In a moment cut from the film. Cut to exterior, house garden, day. The garden of Lena's house where Lena and Kane are sunbathing on the lawn. Lena in a bikini, Kane in shorts. Around them are the remains of an impromptu picnic. They lie side by side looking up at the sky. And in the script, Lena's first line here is nicer than it is in the film. Lena, I love seeing the moon in daytime. It's like God made a mistake, leaving the hall lights on. In the film, Kane will say, it's always so weird seeing it like that in daytime, which is immediately more negative. But before that, this scene, in their bedroom instead of outside in the garden, begins with the end of an awkward silence. We're angled across Lena on Kane. Lena, you're not talking to me. Kane, sorry.
Zoned out.
Lena, thinking about the next mission? Kane, no. I was just looking at the moon.
It's always so weird seeing it like that in the daylight.
Angle across Kane on Lena. Lena,
Like God made a mistake.
Left the hall lights on. Angle across Lena on Kane. Kane,
God didn't make mistakes. It's somewhat key to the whole being a God thing.
Though Lena is in the foreground, out of focus, we can see that she rolls her eyes at this. But Kane's belief in God is interesting because what does Kane think of the shimmer? Would the existence of such an aberration not challenge the fundament of his beliefs? Is that why he goes on his mission to the shimmer, to confront this unnatural thing that challenges his very understanding of the world around him? Lena will acknowledge God in her response, but more grammatically than literally. As a scientist, she might not believe in God at all, but Kane's reference must be dismissed specifically so that she can explain her point scientifically. Lena, pretty sure he does. She does not add a line from the script. I observe his mistakes every day. Kane turns his head toward Lena. Kane, you know he's listening right now, don't you? She laughs scoffingly. Angle across Kane on Lena. Lena, take a cell. Circumvent the Hayflick limit due to prevent senescence. Before Kane makes a joke like he knows what Lena is talking about, but we do not get the scripted line about him being aroused at her patronizing him. Allow me to patronize you. The Hayflick limit, according to Zane Bartlett, 14th November 2014, the Embryo Project Encyclopedia is, quote, a concept that helps to explain the mechanisms behind cellular aging. The concept states that a normal human cell can only replicate and divide 40 to 60 times before it cannot divide anymore, and will break down by programmed cell death, or apoptosis. The concept of the Hayflick limit revived Alex Carroll's earlier theory, which stated that cells can replicate themselves infinitely. Leonard Hayflick developed the concept while at the Weistar Institute in Philadelphia, Pennsylvania in 1965. In his 1974 book, Intrinsic Mutagenesis, Frank McFarlane Burnett named the concept after Hayflick. The concept of the Hayflick limit helped scientists study the effects of cellular aging on human populations from embryonic development to death, including the discovery of the effects of shortening repetitive sequences of DNA, called telomeres, on the ends of chromosomes. Elizabeth Blackburn, Jack Szostak, and Carol Grader received the Nobel Prize in Physiology or Medicine in 2009 for their work on genetic structures related to the Hayflick limit. Carroll, a surgeon in early 20th century France working on cultures of chick heart tissue, argued that cells can infinitely replicate. Carroll claimed that he had been able to have those heart cells replicate in culture for greater than 20 years. His experiments on chick heart tissue supported the theory of infinite replication. Scientists tried to replicate Carroll's work many times, but these repeated experiments never confirmed Carroll's findings. Hayflick worked for the Weisstar Institute in 1961, where he observed that human cells do not replicate infinitely. Hayflick and Paul Moorhead described the phenomenon in a paper titled The Serial Cultivation of Human Diploid Cell Strains. Hayflick's job at the Weisstar Institute was to provide cell cultures to scientists who conducted experiments at the institute. But Hayflick pursued his own research on the effects of viruses in cells. In 1965, Hayflick further detailed the concept of the Hayflick limit in cells in a paper titled The Limited In Vitro Lifetime of Human Diploid Cell Strains. In that article, Hayflick concluded that a cell could complete mitosis or cellular duplication and division only 40 to 60 times before undergoing apoptosis and subsequent death. The conclusion held for many cell types, whether they were adult cells or fetal cells. Hayflick hypothesized that the limited replicative capability of the cell related to aging in cells and consequently to human aging. The publication of Hayflick's experiment disconfirmed Carroll's theory about indefinite cellular replication. Some, such as Harry Rubin at the University of California at Berkeley, argued in the 1990s that the Hayflick limit pertained only to damaged cells. Rubin suggested that cellular damage could result from the cells being in an environment that differed from their original environment in the body, or when researchers subjected the cells to laboratory practices. Regardless of the criticism, other scientists used Hayflick's theory in support of further studies about cellular aging, especially with research in telomeres, which are repetitive sequences of DNA at the ends of chromosomes. Telomeres protect the chromosome from folding in on itself, and they decrease mutations in the DNA. In 1973, Alexei Alovnikov in Russia applied Hayflick's theories of cell death to his studies at the ends of chromosomes that did not replicate themselves during mitosis. He said that the process of cell division ends once the cell cannot replicate the ends of their chromosomes. Although Alovnikov applied Hayflick's theory to his experiments, Alovnikov did not name Hayflick's theory. 1 year later, in 1974, Burnett coined the term Hayflick limit in his work, Intrinsic Mutagenesis. Burnett's work focused on the claim that age was intrinsic to the cells in each species, and that they followed the Hayflick limit, thus establishing a programmed age in which an organism would die. Elizabeth H. Blackburn at the University of California, San Francisco, and Jack W. Sostak at Harvard Medical School also applied Hayflick's theory of cellular aging to their research on the structures of telomeres in 1982, when they cloned and isolated telomeres. In 1989, Breeder and Blackburn further developed the theory of cellular aging to discover the enzyme that replicates telomeres, called telomerase. Breeder and Blackburn found that the presence of telomerase helps cells escape programmed cell death. With theories about the biological mechanisms behind aging, scientists expected that they could create a cure for aging. Hayflick helped found the National Institute on Aging in Bethesda, Maryland in 1974, a branch of the National Institutes of Health in the United States. In 1982, Hayflick also became the president of the Gerontological Society of America, founded in 1945 in New York. Hayflick's role helped to spread the theory of the Hayflick limit and to further counter the theory of cellular immortality as established by Carroll. End quote. Other sources get even more complicated and hard to understand. Essentially, normal cells, under normal conditions, will not replicate forever. Second 44, angle across LENA on KANE. KANE.
I was about to make the exact same point.
In the script, LENA says, "Would you like me to explain what it means?" And KANE says, "I would, because I find it arousing when you patronize me." LENA, it means the cell doesn't grow old, it becomes immortal, keeps dividing, doesn't die. Angle across KANE on LENA. In the script, KANE says, "Don't stop." LENA continued, "We see aging as a natural process, but it's actually a fault in our genes." And time runs out for this minute.
We spoke. What was it we said?
Wordlessly watching, he waits by the window and wonders at the empty place inside.
Annihilation is all we are.
Annihilation.